Every day, we use objects whose shapes are important to their function. An egg carton has depressions designed to cradle eggs so they won't break. A funnel has a wide brim and a narrow neck so liquid can be transferred from one container to another. Inside our bodies, on a much smaller scale, proteins have specialized shapes to tackle their tasks. And while those shapes are much more complex than everyday objects, the structure of a protein offers clues about the role it plays in the body. It may also hold the key to developing new medicines, materials, or diagnostic procedures. Proteins make up our skin and other tissues, carry the oxygen in our blood, help our muscles move, and much more. There are different kinds of proteins. Enzymes speed up biochemical reactions. Scaffolding proteins help cells move and maintain their shapes. Channel proteins let substances flow in and out of the cell. Proteins are made up of long strands of smaller molecules called amino acids. The strands twist, bend, and fold into specific shapes. Scientists want to know more about how that folding happens. Understanding this could provide insight into diseases related to misfolded proteins. Once they're properly folded, proteins' specialized shapes help them to do their jobs. The Y shape of antibodies helps these immune system proteins bind to foreign molecules like bacteria or viruses, while also drawing in other immune system molecules. The grooves or pockets of enzymes help them to hold on to other molecules to speed up biochemical reactions. In an approach called structure-based drug design, scientists use the structure of a protein as a template for identifying new medicines that can block or stimulate a molecule's activity. They start with a computerized model that shows a protein bound to a natural molecule called a substrate, which unlocks a biological action. Here, the protein is the lock and the substrate is the key. To develop certain anti-HIV drugs, scientists targeted the enzyme HIV protease, which is essential to the virus's life cycle. Knowing the protein structure allowed researchers to determine the kinds of molecules that could block the active site and stop HIV protease from functioning. To determine a protein structure, scientists use two main methods: X-ray crystallography and nuclear magnetic resonance spectroscopy. The resulting three-dimensional images can be presented in a variety of styles, including space filling and ribbon diagram. X-ray crystallography shoots a beam of X-rays through a tiny solid crystal made up of trillions of identical protein molecules. The crystal scatters the X-rays onto an electronic detector, similar to those that capture images in a digital camera. The intensities of the scattered X-rays are fed into a computer, which calculates the position of each atom in the crystallized molecule. NMR spectroscopy applies a magnetic field to proteins to see how the natural magnets in some of their atoms respond. The results indicate the position of the atoms in the protein and how they are associated with each other. With new tools and improved techniques, scientists now can determine the structure of increasingly larger molecules and complexes, including the protein-making ribosome. They are also determining structures more quickly than ever before, dramatically increasing our understanding of their roles in the body and spurring advances in medicine.